Dear students, this is our 16th lecture on bioorganic chemistry and second lecture on enzyme catalyzed reactions. In the first lecture we have seen some basic concepts, and in the present lecture we will be discussing addition and elimination reactions which are catalyzed by some enzymes. Now these addition elimination reactions, basically they are simultaneous addition to unsaturated carbon-carbon double bond followed by elimination of inorganic phosphate molecule in presence of enzyme. Then these types of reactions, they are called as addition elimination reactions. And the most common example of such type of addition elimination reaction is synthesis of EPSP, that is 5-enol pyruvyl shikimate 3 phosphate, and this is formed from the reaction of shikimate 3 phosphate with phosphoenolpyruvate in presence of enzyme 5-enol pyruvyl shikimate 3 phosphate synthesis. So when these two they combine in the presence of this enzyme, and addition elimination reaction take place to give us this type of EPSP product. EPSP means 5-enol pyruvyl shikimate 3 phosphate is formed. Now the mechanism of this reaction, there are 4 types of mechanism which have been given for this. The first type of mechanism given is a concerted addition elimination mechanism. So this is concerted addition elimination mechanism. Here what happens is that the lone pair present on this hydroxyl group act as a nucleophile and attacks this electrophilic carbon-carbon double bond, and this carbon abstracts a proton from the active site of the enzyme which is acting as an acid, and this is the addition step of the reaction, and this addition step then will undergo elimination where this active site of the enzyme will act as a base, and this will abstract the proton back which was given in the first step. This is abstracted back, and then this goes to this place to create carbon-carbon double bond, and a molecule of hydrogen phosphate is removed from this place, and this is how these two substrates they can be converted into EPSP. So 5-enol pyruvyl shikimate 3 phosphate is formed by this type of concerted addition elimination mechanism. Then second mechanism is a stepwise addition elimination. Here this is a concerted mechanism. Here the product is formed in different steps. In the first step, the active site on the enzyme gives a proton to the phosphoenolpyruvate, and a carbocation is generated by addition of this proton to the carbon-carbon double bond, and this type of carbocation is formed, and this carbocation is attacked by the nucleophilic hydroxyl group of the shikimate 3 phosphate to give us this type of intermediate, where now the positive charge will be present on the oxygen, and we know that positive charge on oxygen, being an electronegatory element, is not a very stable combination. So then active site of the enzyme will abstract one proton to neutralize this oxygen, and along with the neutralization of this oxygen, this hydrogen phosphate molecule will be removed from this place, and we will get this type of carbocation. So the basically the positive charge here is on oxygen, which is less stable, and here in this intermediate the positive charge is on the carbon. So carbocation is formed. Then the active site of present on the enzyme will abstract one proton from this carbon, and we will get our product by elimination of the proton. So this is how a stepwise addition elimination will take place, and we will get our product in the same way. Then third mechanism is a covalent concerted mechanism. So here a covalent intermediate is formed between the enzyme and the substrate. So this nucleophile present on the active site of the enzyme will attack this electrophilic carbon, and one proton will be abstracted from another active site of the enzyme to this carbon. So basically addition will take place across this carbon-carbon double bond, and we will get an intermediate of this type where enzyme is bound to the substrate. So this is a covalent enzyme substrate complex which is formed, and this covalent enzyme substrate complex will then interact with another substrate, that is shikimate 3 phosphate, and this hydroxyl will attack as a nucleophile at this place with the elimination of the phosphate group from here. And again this is another type of intermediate where also we both the substrates, they are bound to the enzyme in a covalent fashion, and then another active site of the enzyme will abstract this proton, and this will create a carbon-carbon double bond with the removal of this covalent bond present on the enzyme. So this is how we will get our product by covalent concerted mechanism. Then fourth mechanism is covalent stepwise mechanism. So similarly as we have seen in the second mechanism, here also we will get the product in a stepwise manner, but only thing is here also a covalent intermediate between substrate and enzyme will be formed in the first step. So in the first step again, the nucleophile present on the active site of the enzyme will attack this carbon. This proton will be abstracted from another active site of the enzyme, and we will get this type of enzyme substrate complex as we have seen in covalent concerted mechanism. Then this will be in equilibrium with this, because this proton which was abstracted will be again added to the carbon-carbon double bond through the active site of the enzyme, and we will get a carbocation of this type, but this carbocation is also an enzyme substrate carbocation where there is a covalent bond between the substrate and the enzyme. Enzyme substrate covalent intermediate is present, and then this is attacked by the second substrate in the next step, and we will get this type of covalent intermediate which will rearrange to give us the product in a stepwise manner. But in all the intermediates we are having an enzyme substrate complex which is covalent in nature. That is why this mechanism is called as covalent stepwise mechanism. And then isomerization reactions, these are those reactions where enolic intermediates, they involve 1,3 hydrogen shift. So like this, the simultaneous acid base enzyme catalysis is the mechanism for such type of reactions, that one of the active site act as a base which will abstract proton from the water, and this will increase the nucleophilicity of the water. This will attack the electrophilic carbon, and the proton will be provided by the another active site present on the enzyme to the oxygen, and this is how ketones, they will be converted into enols like this. So in these types of reactions, which are called as isomerization reactions, aldehydes, ketones, thioesters and carboxylic acids, they undergo enzymatic isomerization reactions, and you can see the general mechanism of these types of reactions. The alpha proton, which are present at the carbon adjacent to the carbonyl group, these are very acidic protons because of the presence of this carbonyl group, and because of their high acidity they can be abstracted very easily by the active site of on the enzyme which is acting as a base. So by abstraction of this proton, this will create a carbon-carbon double bond, and this will abstract a proton from another active site of the enzyme, and we will get this type of intermediate. And then this can again undergo rearrangement to give us alpha beta unsaturated compound. So such type of reactions, they are called as isomerization reactions. Then mechanism of these types of beta elimination reactions, in case of aldehydes, ketones and thioesters, they show that syn stereochemistry is formed, and only one base is involved in the proton transfer and elimination. You can see over here, in the previous example we have seen that the product formed will be having syn stereochemistry and only one base is involved in the reaction. But if we use carboxylic acid, then the reaction will proceed with anti-stereochemistry, and two bases they are involved from the enzyme in proton transfer. So you can see over here, this is the one base which is used. With this will abstract this proton, and then this will act as an acid with the elimination of this X, and this third one will be acting as again as I said to transfer this proton to the oxygen. This type of intermediate will be formed, and then in the presence of metal ions, this negative charge is neutralized, and we will get alpha beta unsaturated carboxylic acids as the product in these types of isomerization reactions. Then this is a typical example of isomerization reactions, that how pro or hydrogen is removed from the substrate. So you can see over here, in this reaction, this is HS and this hydrogen is having R. So base will remove this R from this, and this through anti configuration will be converted into Z isomer. So anti conformation means this carbonyl is at this position and carboxylic acid is at this position, whereas in syn this carboxylic acid is present over here and keto is present over here when HR is removed by the base, and we will get E isomer as the product. So how these two different products, that is anti and syn, they are formed can be explained by this mechanism. Then next is elimination reactions. Elimination reactions, they generally give unsaturated products by removal of alpha hydrogen from the substrate, followed by elimination of the other group at the beta position. So the other group may be hydroxide ion or phosphate ion in biochemical reactions, and these elimination reactions, they also proceed either by anti mechanism or syn mechanism, and these are the general mechanism as we see in chemistry, that either this can involve a carbanion E1CV mechanism or concerted E2 mechanism or carbocation mechanism for elimination reactions. So E1CV mechanism, first conjugate base is formed like this by removal of this proton by the nucleophile, and then removal of this beta group to give us unsaturated product. And here the same reaction take place in a concerted elimination mechanism, which is called as E2 mechanism. Or third one is E1 mechanism, where first a carbocation is formed, and then that carbocation undergoes elimination of the proton to give us the same product. And example is elimination of water, and elimination of water follows anti mechanism, and it takes place in presence of enzyme dehydratase. So dehydratase, they can produce unsaturated compounds by either of these three mechanisms. In the enzymatic reactions, these three mechanisms are not the only mechanism which are followed, but in addition, some cofactors, they are also used by the enzymes for these types of elimination reactions. The most commonly used cofactors, they are beta-ions or NAD plus or iron-sulfur clusters. For example, an important reaction in glycolysis is reversible elimination of water from this substrate to phosphodiglyceric acid, which is called as 2-PGA, to produce phosphoenolpyruvate, which is catalyzed by enolase enzyme. And these two protons present in the product, that is phosphoenolpyruvate, they can be differentiated on the basis of NMR spectroscopy. For example, this proton A will absorb at 5.33 ppm, whereas this proton B will absorb at 5.15 ppm. So on the basis of NMR, we can differentiate whether an anti mechanism or syn mechanism is involved in these types of reactions. For example, elimination proceeds with an anti mechanism. This is proved from the fact that as we have seen in the previous slide, that there are two multiplets, one at 5.33 ppm and other at 5.15 ppm. When enolase dehydratase is used as an enzyme, and if one of the hydrogen is replaced by deuterium, then in the product we observe only one signal, that is at 5.14 ppm, which indicates that deuterium in 2-PGA lies at trans to the phosphate group. So this is phosphate group, and deuterium lies trans to this phosphate group. Only then this type of phosphoenolpyruvate can be formed, where this HB is present at 5.14. So which proves that elimination of hydroxide at C3 is anti-elimination. Then another example is biosynthesis of 6-deoxyhexoses. Here if we start with nucleoside diphosphohexose A, this one, where R is some nucleoside diphosphate. In the presence of enzyme nucleoside diphosphohexose 4,6 dehydratase, this is converted into this type of nucleoside diphospho 4-keto D-glucose, and this is in equilibrium with nucleoside diphospho 4-keto D5,6 glucosine, where dehydration at this these two carbons takes place, and we will have this type of methylene group which will undergo rearrangement in the presence of NADH, and one hydrogen from NADH is shifted to this carbon-carbon double bond to become methyl. So overall here in this conversion, elimination dehydration of the glucose or hexose takes place. Similarly, in case of aromatic amino acids and other aromatic metabolites in bacteria or fungi and plants, they are biosynthesized from crosinate. So crosinate is formed in the last step of shikimic acid pathway by elimination of this phosphate from EPSP 5-enol pyruvyl shikimate 3 phosphate in the presence of enzyme crosinate synthesis. So basically this is again an example of elimination reaction, but here the elimination of the phosphate is taking place to give us this as the product in an enzyme catalyzed reaction. The mechanism of this reaction is that in the first step, the nucleophilic active site present on the enzyme will attack this carbon-carbon double bond, and shifting of this carbon-carbon double bond will be followed by elimination of this phosphate group, and this will give us a covalent intermediate between the substrate and the enzyme. So covalent enzyme substrate complex is formed in this manner. Then this may undergo E2 elimination where the active site in the form of base present on the enzyme will abstract this proton which is adjacent to the carboxylic acid, and this will undergo E2 elimination. This will create a carbon-carbon double bond, and this enzyme substrate covalent bond will be broken between the two, and we will get crosinate as the product. This is one pathway which can be followed, that is E2 mechanism. Another mechanism is that in the first instance this bond breaks, and we will have a positive charge on this, a carbocation is formed like this, and then in the second step the active site of the enzyme which is acting as a base will abstract this proton, and we will get the crosinate as the product. So there are two mechanisms, one is E2 mechanism and other is E1 mechanism, which is followed in the 1,4 trans elimination of pro R hydrogen from the substrate. Then another example of anti-elimination is elimination of ammonia from histidine to give us urocanic acid, and this conversion of histidine to urocanic acid is catalyzed by enzyme histidine ammonia lyase, and this is again an elimination reaction where deuterium from here and ammonia from here is eliminated to create a carbon-carbon double bond, and this again is an example of anti-elimination. Then another example of syn-elimination is that 3-dehydroquinate is converted into 3-dehydroshikimate in the presence of enzyme 3-dehydroquinate dehydratase. So here the active site of the enzyme, which is in the form of lysine amino group, will act as an active functional group which will create a carbon nitrogen double bond, or shift bases will be formed in the initial reaction with the substrate. So enzyme substrate complex is formed like this in the elimination of water, oxygen from here, H2 from here. So enzyme substrate complex of this type, a equivalent shift base will be formed. Then another active site present as a base will abstract this proton HR from the adjacent carbon with the creation of carbon-carbon double bond at this place, and this carbon-nitrogen double bond will be converted into carbon-nitrogen single bond, and then this nucleophilic nitrogen will attack at this place. Then this carbon-carbon double bond will be shifted to this place with the elimination of water by the active site, which is acting as an acid. This will give proton to this hydroxide to give us water, and this will active site will again become a basic active site in nature, and then this carbon nitrogen double bond will undergo hydrolysis to give us the product. So this is the mechanism how a syn elimination, which follows E1CB mechanism, or a shift base is formed in the first step, is formed in the elimination reactions.